Good afternoon. I think let's get started. Now, welcome back to Track4, Science, Technology, Innovation. And thanks for sponsor this afternoon's lunch. My name is Jing Wen. I'm your room hosting for this afternoon's section. I am a lab director at Regenerative. Just a reminder, I would be also welcome those who join remotely through a live stream. And remember to download our mobile app to use some of the highlighted features like live Q&A. You can submit your question during or after the meeting. For this afternoon section, the promise of regenerative medicine, cell therapy, and tissue engineering. We are joined by the great panel, Maury. So Maury will be this afternoon's moderator, so I will pass to Maury.
Okay,
thank you.
All right, thank you very much. And welcome back, everybody, from lunch. So we're going to jump right into this. So yes, my name is Maury Ruffin. I'm the managing director of the Alliance for Regenerative Medicine and one of its co-founders. And here, let me make sure we have the. So the way we're going to do this afternoon is I'm going to give a little bit of an overview, and then we're going to go to each of the panelists, and they're going to each address really a different aspect of cell therapy in the context of rare genetic disease. And I think hopefully through that presentation, this will be, I think, a nice overview of what we see as the outlook for cell therapy in treating genetic disease. And then we'll have hopefully some time for questions after that. I've got a few follow-up questions that I'm going to provide as the moderator. It's my prerogative. But hopefully we'll have enough time to pass the mic around and get a little bit of a conversation going. Before we start, though, I did want to do a little shout out and a thank you to Nicole for inviting me here. This is really a tremendous event. I think this has a really important place in the rare disease community and dovetails, I think, very nicely with the work that we're doing at the Alliance for Regenerative Medicine. And we've had a strong working relationship and partnership with the team at Global Genes really now for the last 3 or 4 years. I wanted to point out Chelsea Hathaway. Chelsea, if you could raise your hand. She is in charge of all of our patient outreach work at the Alliance. And this is a very fundamental part of what we do and who we are as an organization. We are not a trade organization. We are, as the name suggests, an alliance. We work with patient groups. We work with major research institutions, and you'll hear about some of them today, in addition to companies and doctors and physicians and really the whole community of people involved in developing advanced therapies to treat rare genetic disease. So we couldn't do it without the partnership that we have with groups like Global Genes. Oops. Am I doing this the right way? I'm pressing the green button. Oh, there we go. Okay, let's go back then. So just a couple words on ARM's mission. So I talked a little bit about the Alliance for Regenerative Medicine. We are primarily an advocacy organization, and what we aim to do really is to foster research, development of these technologies, hopefully investment in this sector, but ultimately for the purpose of delivering this to the patients. And we do this through a membership which is now about 300 companies and organizations, really representing a large group of stakeholders. I just named a few of them previously, but it really is a collective effort. And so much of what we're doing is really focused on improving the standard of care for patients. And we really believe that these technologies fundamentally can change how we treat patients. And I think that's what you're going to hear about today. I know there's a session after this that will focus on gene therapy. We're really going to be focusing on cell therapy today. But this is really looking at how hopefully we can modify disease. We can hopefully find cures for many of these indications, especially a lot of the monogenic disorders we'll be talking about. But this is really a different type of medicine than most of us are familiar with. And I think hopefully you'll be able to understand a little bit more about that as we talk about the potential of cells and cell therapy and gene therapy. So this session, and then the session after this, will focus on gene therapy, but this session is going to focus, as I said, predominantly on cell therapy. And cell therapy, in the context that we're going to be talking about it, is really cells as delivery vehicles, vehicles for genes, for proteins, ways to modify tissues and cells in the body. And there are a number of different types of cells that allow us to do that. And we're going to talk about some of them. Another important component of this, and you've heard about this in prior sessions too, a number of these are autologous therapies, and John's going to talk about some of those. Linda's been doing both autologous and allogeneic therapies. And then we're going to talk about other platforms there too. Gene therapy and genetically modified cells are also a very important part of this, and we'll touch on that through the discussion and the presentations. But just a little bit of kind of an overview of what cell therapy is. So we talk about a number of different cell types. And at the session just before lunch, they were talking about induced pluripotent stem cells. And Tom Novak is going to talk about that as he presents the work of CDI. But we also are going to talk about the use of a number of other different types of cells. So we're going to talk about adult progenitor cells, and we're going to talk about the use of MSCs, but also there's cells that are pluripotent ESCs, and you probably heard discussions about those in some of the other sessions here at the meeting. But ultimately, again, as I said earlier, this is really about how these cells can be used to deliver a therapy. And in the context of rare disease, so the purpose of this meeting, and I think you'll see here, I mean, we see tremendous potential for this technology here. As we know, about 80% of genetic diseases involve some sort of genetic component, or rare disease involve some sort of genetic component, many of them monogenic. And I think this really presents us with an opportunity to focus in on the disease itself, the pathology of the disease, and how that can either be treated or corrected. And this can be done, as we said, using genes, but cells are a really important component of this. And so the pipeline for cell therapy and gene therapies is really quite large. And at ARM, we track the clinical development pipeline on a regular basis. We have our annual State of the Industry briefing, which we do every year in January, and then we put out quarterly reports, and we track the progress of these therapies through the clinic. So what we look at now is a very robust pipeline with almost 900 clinical trials ongoing, and this is in cell and gene therapy. I would venture to say, and we haven't counted up all of them exactly, but I would say about close to 50% of these are probably directed at some form of rare genetic disorder. So a big part of this pipeline is focused on rare disease. And in terms of indication, it really covers the entire waterfront. I mean, obviously oncology garners a lot of attention, and there is a significant number of trials in the oncology space. But as you'll hear about today, cardiovascular disease, dermatology, CNS, neuromuscular diseases, I mean, all of these are areas where there's a tremendous amount of work going on and a lot of potential and promise. And so as I wrap up and hand over the podium here to the panelists, I just wanted to mention a few reasons for optimism in this space. You know, I think one of the challenges and one of the concerns has always been, you know, are there real safety issues around cell therapy? And obviously we have to take this very seriously. I think we can say with some level of confidence that the number of adverse events we have seen around a number of these programs in therapeutic development are really low. So I think in terms of the safety profile for these therapies, I think there's real reason for optimism. There's been a lot of discussion in Washington recently about this sector, and as many of you know, the 21st Century Cures legislation was passed in December, and there was a very important provision in that legislation which creates an accelerated pathway for approval of cell therapies in regenerative medicine. And this was a collective effort. ARM led the effort, but I think it's really an important signal for this sector that the FDA recognizes the importance of this. Certainly Congress did. And I think for people developing therapies, there's now a much clearer pathway to get them through the clinic and to the patients. And then just a couple of other things to mention. Obviously, recent approvals in the last session before lunch, some of these were mentioned, but these are really the first cell therapies that have been approved. Strimvelis, and these involve gene therapy components to them as well, but the delivery vehicle in these cases are really cells. And as we said, I think this pipeline is robust, so real reason for optimism. And then the last point I will make is that the FDA leadership, I think, is very strong right now. Dr. Gottlieb is somebody many of us know in Washington, done a lot of work with. He came out just at the end of August with a policy statement on regenerative medicine that was very strong. Where he talks about the potential in regenerative medicine, cell and gene therapy, to significantly alter the course of disease, and about the FDA's commitment to making sure we get these therapies through the clinic and to the patients as quickly as possible. So a lot of reasons for optimism in the cell therapy space. So what I'd like to do now is turn this over to our panelists. Let me just quickly introduce them to you. So sitting to my immediate right is John Maslowski. John is the CEO of FiberCell. And sitting next to him is Linda Marban. She's the CEO of Capricorn. And then last but not least is Tom Novak from CDI. And he's going to pick up at the end and talk about iPSCs, really, both in the research context, but also in the as potential for developing new therapeutics. So why don't we begin with you, John? Thanks.
For those of you in the back, there are some seats up front here, if you don't mind sitting on the front row.
Is this kittens? All right, do they have slides ready? And the next. Okay, great. All right. All right, thanks, Maury, for the introduction, and thank you, Nicole and Global Genes, for inviting us. We're truly honored to be part of this panel today. And what I'm going to do is give you just a very short overview of FiberCell and what rare diseases and what we're targeting and our methods for doing so, which are in rare skin diseases. That's what we're really focused on, using a cell called a fibroblast. And if you're not familiar, a fibroblast is a cell that makes up most of your tissue in your body, and it's part of the dermal layer of your skin, which is the layer underneath the second layer of your skin. And we use these cells to, find genes of interest to target specific diseases. And I'll be talking about some of those in these next few slides, about 3 or 4 slides. So this is just a quick at-a-glance of our company and what kind of platform we're looking at. Again, using this autologous fibroblast, so autologous from the cell, this skin cell, to take them from the patient, grow them up into significant populations, and then use viral vectors to insert genes of interest in them, and then have that same product then delivered back to the patient so that it is their own cells. And we have our lead product is called FCX007, and it's a fibroblast-based therapy for recessive dystrophic epidermolysis bullosa. And if you're not familiar with the disorder, it's a rare blistering disease that's caused by a mutation in just one protein, type 7 collagen. And I'll talk a little bit more about that in a moment. This is actually in a phase 1 program now. We've dosed 3 patients, and we actually have a data readout coming up shortly in the next few weeks. And we have a number of, Maury just talked about some of these new drug designations and vehicles that the FDA uses to help us, move through our review with them faster, have other access to the FDA to make sure they can have questions answered. And these here are an example of a few that FCX007 has: orphan drug disease, rare pediatric disease designation, and fast track. So there are a lot of these great programs FDA are using to help us learn more through our process, move through these steps with them quicker, and really get access to them for questions and for back and forth. We have a second program in another rare disease called localized scleroderma. So you might be familiar with scleroderma and hold, which is a hardening of the skin or of organ tissue that creates a lot of problems, especially in the severe systemic type. There's also skin-based ones called localized scleroderma. I'll talk a little bit more about how that affects patients and what we're doing to try to work with that as well, targeting a gene to break up that extra sclerosis. And then we have other larger indications in arthritis, but today I'm going to be focusing on the top two. So this is just a cartoon just basically of how we manufacture these therapies. And what we do is we take small biopsy punches from our patients. They're then grown up in our cGMP labs into populations. And then we use viral vector. If you're familiar with that technology, this has just been a way to introduce genes of interest that we create to cells. And the technology for this, and I'm sure we'll discuss it in the panel, the safety and all of the technology around this has grown exponentially over the last 20 years. And that we have just really wonderful tools now for actually genetically modifying these cells and hitting targets we really want to for disease. So we introduce this into our manufacturing process early on, and then we actually can grow those cells with that gene and continue to make more and more copies of it. And then those cells are cryopreserved, so we actually freeze them down, and then when the patient's ready for dosing, we take those out, we thaw them and wash them, and then it's shipped to the investigator site for injection. So just a little bit about each of the diseases that we're looking to treat with these cells. The first one being RDEB, and again, if you are familiar, I apologize for going over the disease again, but it's really, again, one protein. It's amazing how much havoc a mutation in one protein can occur. If you look at the structure of the skin on the left there, that's our normal human skin, and there's these sort of arcing fibrils that connect in between the layers of the dermis and the epidermis that bind them together. And that's called an anchoring fibril. You think of it sort of like Velcro, sort of have the hoop, and then you've got like the kind of other part of the Velcro that it binds to. These bind the layers together, they wrap around your collagen fibers in your lower skin, and then that creates this anchoring fibril structure. That is made up of type 7 collagen. So when you unfortunately have a mutation in that disease, in that particular protein, the gene for it, you either produce poorly formed versions of it or you don't make it at all. So you then end up with the situation on the right where, a little bit of lateral friction on the skin creates a void, and then it creates a blister, and then it creates a wound, and then you have these just terrible issues with chronic wounding all through these patients' life. It's devastating. It's progressive. It's got a high mortality rate. I mean, if you have all this wounding and rewounding, squamous cell carcinoma becomes a very large issue for these patients, and they have a high mortality rate, 76% by the time they're in their 30s. So as you can see, it's a lot of suffering, and right now there are no currently approved therapies. Right now it's really palliative care of the wounds. Patients and families are paying up to $10,000 a month just in bandaging and antibiotics alone, never mind all the surgeries, finger separation, esophageal dilatations. So, what we're really hoping is that if we can deliver this protein properly to the skin, we can help keep it together and help them with their wounding. As you can see from the epidemiology, it is very rare. There is a larger subtype, but the RDEB subtype we're targeting is about 1,100 to 2,500 patients in the U.S. So we're in a clinical trial on this now. The goal of the trial is to obviously assess the safety of our product, but we're also going to be looking at some other endpoints, including looking at rates of wound healing and looking at some protein expression in the skin, just trying to see if we're seeing any functionality of it through these patients. We're targeting 6 patients in phase 1, 6 in phase 2. We're doing adults in phase 1. There aren't many of them, but we do have great work that we do at Stanford University that can find these patients and help us treat them. And then we're looking to add some pediatric populations into the phase 2 portion of the process. So we've dosed some patients, and we're looking to continue that, and then also going to be reading out some data in the next few weeks just to get an interim look at where we are. And then lastly, just wanted to touch on scleroderma. This is a program that we have that is in what we call the preclinical phase. So we're still looking at this in the lab. We're looking at this in animal studies just to make sure it's safe and that we see the product is working properly in those models before we introduce them into humans. So this disease, if you've heard of it, is a thickening of the skin from an autoimmune reaction that occurs just sort of randomly around the body when you have the disorder. In children, it tends to occur from 5 to 15, and it occurs in a linear form, most likely, where it can pass over joints It can be very deep, and it actually can form on the forehead, and all these lead into developmental issues, neurological issues, so it's not just a cosmetic issue. You may think that it sounds like plaques and they sound like they're bad, but really, do we need a gene therapy for it? It does have a lot of morbidity, especially in children because of development. Right now, current treatments are really corticosteroids, methotrexate. They've tried some light therapy, nothing approved by the FDA. So what we're looking to do here is treat these patients, about 90,000, which are considered to have the severe form of it, and inject these cells locally, have the protein that's produced to sort of break up that sclerotic region and try to return their normal functionality and hopefully arrest any kind of impact it would have on development. Okay, so those are just some references. So thank you very much. Just a short overview of our programs, and looking forward to hearing more from the panel.
Good afternoon. My name is Linda Marban. I'm the CEO of a company called Capricorn Therapeutics. We are located just up the road in Los Angeles, so it was an easy trip down here for me today. And while they load my slides, I'll say that we have been spending, I'm sorry. Oh, I just do it myself? Okay. I'm sorry, I'm used to people taking care of me. Which is totally not true, by the way. We're a publicly traded company. This is a standard forward-looking statement. Our symbol is CAPR, if you're interested in reading about what we're doing now and also in the future. So we have a cell that we call CAP1002. This is a cell that is made from heart tissue, but we use it specifically to treat all types of muscles. And it's specifically designed to work in areas of inflammation, which is what occurs when you have some types of diseases, especially genetic diseases where proteins aren't made correctly. And then also fibrosis, which is the scarring that occurs from chronic inflammation. One of the things that's really interesting about our cells is that we understand how they're working in terms of attacking the inflammatory processes. And basically what they do is they calm down inflammation. When you have injury to a tissue, you have something that comes to the scene called a macrophage. And a macrophage is the immune system's way of saying there's somebody here that we don't like, and they're angry. They want to fight and get that bad thing, whatever it is, out of there. And obviously, inflammation has the good side because it gets a lot of bad stuff out of your body, but it also has the bad side, which is that it can sometimes be too strong for what the situation calls for, and especially in a certain type of genetic disease where your body's fighting really hard anyway. The last thing it needs to do is fight against itself. So what our cells do, and they do it via these things called exosomes, and exosomes are made by our cells, so it's the mechanism of action. It's these tiny little bubbles that are released and change the activity of a cell. What the exosomes do is they calm down the macrophages. We've published this in the scientific literature, and I won't go through the details, but we've shown in very careful studies, both in cells, dishes, and then also in animals, we've shown that these cells work by calming down the macrophages and shifting them from their angry phenotype to their calm phenotype, which is called M2. And that basically are the macrophages saying, all good here, don't worry about it, everything's okay, and it allows normal physiologic processes to overcome the pathology. So it's very, very important to find that balance in inflammation. So the first study that we did in rare diseases, let me just say the company's been around for about 10 years. We started off as an autologous company, so we made cells from the patient's heart tissues and infused it back into patients that had had heart attacks. So our cells have been in several hundred patients at this point, and we know that they're very, very safe. And they were used to treat patients with heart attacks. And what we found with our autologous cells is that it reduced the amount of damage done to the heart by the heart attack. So it actually reduced the amount of scar. Well, when you start thinking about diseases of scarring, you start to think about all kinds of opportunities in the rare disease space, because that's primarily what's impacted in many of these diseases of genetic origin. And so we decided to look at how our cells worked in Duchenne muscular dystrophy, which I think everybody in this room knows what Duchenne is. We've completed our first clinical trial. It was a 25-patient trial. It was called the HOPE trial. We are targeting the patients that are farthest along in the disease process. So we treated those patients that are primarily non-ambulant. 80% of our patients were non-ambulant, and the average age of enrollment was 18. So this is a fabulous patient population because they're very eager for therapeutics. They really don't have too many other options available to them at that time. We recruited the patients, 25 patients in 6 months at 3 centers, and we were proud to say that we were at some pretty amazing sites for Duchenne care. And then they were randomized either to receive our cell, and in this trial they got 75 million cells, or to the usual care group. Now, remember I told you that we founded our company based on autologous treatment of heart disease. Well, what we found along our journey of understanding the mechanism of action is, one, we could use donor cells. We don't have to take cells from a person and do anything to it and give it back to them, so they don't have to undergo 2 procedures. And 2, we started this business, we thought that we had to deliver the cells directly to the heart to treat the heart. So the young men in Hope had to come into the cardiac catheterization lab and got the cells infused into their hearts directly. And it was very safe. They did great. One of our patients was Snapchatting along the way so his friends could see what he was up to. And then we brought them back in to study them over time. And what you see is that we studied their heart function at 6 months and at 12 months, but we also studied skeletal muscle function along the way. So the cells were very safe, as I mentioned. We were able to get through this process without any real problems. The kids didn't mind coming into the cath lab. But what we learned is that we don't need to deliver the cells to the heart anymore. We realized that if you deliver them into the circulation of the heart, they really just go throughout the body anyway. So what we learned, which was very important, is that we can now just deliver them into any blood vessel. And you'll see that in our next clinical trial, the kids will be just getting the cells through an IV infusion. So we thank our patients for going through this process so we could learn this and now provide an even simpler way for them to access our therapy. This was the first tranche of data that we got from the HOPE trial. Red were the treated patients and blue were the usual care patients enrolled at the same time. And what you can see is this is a measure of heart function measured by cardiac MRI. And you can see that the heart function in these boys and young men improved drastically when they were treated with the cells as compared to the natural history. Let me just say improved in Duchenne muscular dystrophy is an almost unheard of word, right? Stabilization is what the clinicians mostly are gunning for, and even the patients themselves. So the fact that we actually saw an improvement was really something that was highly motivating to us, to the patients, the advocacy community, the clinicians, and the FDA, thank goodness. We also, in addition to studying the heart, because we knew that cardiac is muscle, so we were using a muscle progenitor cell, we also studied the muscular function in terms of skeletal muscle. And the time is short today, so I won't spend a lot of time on this, but we used a very well-tested, validated measure called the performance of the upper limb, which was actually developed by the patients, the clinicians, the advocacy community, in order to measure skeletal muscle function primarily in those that are no longer able to ambulate, but also are associated with activities of daily living. So for instance, hand to mouth with or without weights, stacking light and heavy cans, it means that you can do this, right? You can actually take a drink of water or coffee or whatever it is that you want to do. So these are very impactful tests. And what we found, which was really tremendously exciting to us and to the patients and our clinician leaders, is that we saw improvement in skeletal muscle function as well as cardiac muscle function. So you can see here that in the performance of the upper limb, the patients that were treated with the cells did much better at 6 weeks, were still significantly improved at 3 months, and that effect was beginning to drop off by 6 months. So the therapy gave us a gift as well. It showed us when we need to redose. So now we have a picture, right? We don't need to inject into the heart anymore. We can inject intravascularly, typical IV, and we know that we have to redose every 3 months so that we can continue to recapitulate this effect. And we took this data to the FDA just this summer, and we'll be moving forward with what we hope will be a registration study in Duchenne using our cells starting later this year, early next year, so look for that. So basically, I just went through all of the conclusions and talked a lot about what the cells were doing and how they do it. This is an off-the-shelf product. It's frozen. When a clinician would like to dose a patient, they call the distribution facility, and it can be thawed on the bench and delivered safely into an IV, and that's how the new therapies will be given. And this is sort of an example of what the new clinical trial will be looking like. We are going to call the trial HOPE 2. The first trial was called HOPE 1. I'll tell you that we spent about 6 months at our home team trying to decide what to call it, and we kept coming back to HOPE 2 because that's what we want to do. We want to provide hope to these patients, hope to their families, and hopefully will make them better and help them have not only longer but a better quality of life. And we expect to begin that trial later this year. So I want to thank you. Thank you for coming today. I know many of you are parents and families dealing with significant issues, and my hat's off to you. I've spent a lot of time in this community now, and I know what a tremendous lift this is. And I'll just also say that I'm a mother of 5 and a grandma of 4, so I know exactly what you're dealing with your families, and I hope that I can help make your families better. Thank you.
Try to go fast as well. Hi, I'm Tom Novak. I work for a company called Cellular Dynamics. We are a company that produces cells from stem cells. So regardless of whether you're treating a rare disease or a prevalent disease, there are really 2 things that you need if you're interested in cell therapy or tissue engineering. Well, in addition to money and luck and all the other things that are required, you need cells and you need scaffolds. And so scaffolds are either sheets or 3-dimensional structures that you may need to embed cells in. You don't need that for all types of therapies, but you do for some. We don't do that. CDI is focused on producing high-quality cells to a reproducible specification. We started out to do it as research tools, and we're now moving into the therapy space. So when we were founded in 2004, which here, our founder, Jamie Thompson, who had isolated the first human embryonic stem cells, felt that cell therapy was probably a decade in the future, but that there was an immediate need for high-quality human cells for drug discovery, standard small molecule pharmaceutical discovery, to replace animal models and human cell lines and donor tissue that wasn't reproducible. And that's what we started doing. So we've been around now for about 13 years. We were purchased by Fujifilm of Japan. It's a long story why. We can talk about that after, as to why a film company is interested in stem cells. Based in Madison, Wisconsin, we have a lot of experience in stem cells. It's a very new field, human stem cells, and yet we have hundreds of years of experience in our employees. And in last September, we announced that we were splitting into 2 divisions, a life sciences division and a cell therapy division. And as I said, we make cells to a specification. So what we do, what Cellular Dynamics expertise is, 3 areas. It's reprogramming. So we can take adult cells, skin cells, or blood cells, and by adding a small number of genes, we convert them back to a stem cell. From that stem cell, we can make any cell in the body that we want. So that's called reprogramming. It won Shinya Yamanaka the Nobel Prize in 2012. And it's a phenomenal technology because it avoids having to use embryonic stem cells, which obviously have a lot of moral, ethical, and political baggage with them. But more importantly, we can take them from adults with known diseases. So if that disease recapitulates in the cells we differentiate, like heart cells, you can actually use it for drug discovery, another way to treat patients. So it's phenomenal technology. We can also do genome engineering. So you've probably heard a lot about CRISPR-Cas9. It's been in the news quite a bit. One could take stem cells. It's a reproducible source. We can proliferate them indefinitely. We can engineer them. So we can add genes. We can correct mutations. We can alter them in some way that makes them physiologically and therapeutically more useful. And then we can also then differentiate them, and we make cells from them. We make a variety of cell types, and this is what we currently produce, and these are for research purposes. So you can see a lot of various neuronal cell types. We make various heart cells, liver cells, cells that right now have been very popular for drug discovery. But by having these cells and being able to make them so that the cells you buy today are identical to the cells you bought 5 years ago, and identical to the cells you'll buy 5 years from now, it allows companies to use these as a baseline resource for drug discovery, for safety assessment, and for discovering new drugs. But because we can make these cells in a very reproducible fashion, we thought, well, we should be starting to think about making these for cell therapy purposes. And this is why our success has been based on the fact that we make these cells. They have normal human function. We can scale them up. That's what's key. And that we can control their properties. And again, we can show that they're very reproducible from batch to batch. And that's not only necessary for research purposes, but even more important when we get into the clinic, where patient safety is more important than customer satisfaction. So we use a blend. Our quality systems are a blend of GMP and ISO, and GMP is for patient safety. ISO is more for customer satisfaction. The research tools are more of an ISO, but GMP is required when we get into the clinic. So we have a blend of that, and now we're starting to move in that direction. And if you could start the movie, Mike, I just wanted to show, these are heart cells that we produce. These are in culture, so these are from a human blood cell that we converted to a stem cell and then made into heart cells. You can see they're beating in culture. Soon you're going to see, here there's the same field, but on the right we've added a drug that causes them to beat twice as fast as the cells on the left. So you can see these cells show normal physiology. This is what you would see in a patient if you gave them this drug, and you can see it on these cells here. And then this is just a time lapse of some of these neurons that we've produced, and you can see that they're putting out processes. So these dendritic processes, they're forming synapses, connections with other neurons, and over time they form a very dense neuronal network. So these would be useful for studying neurodegenerative diseases. They could be used for studying psychiatric diseases, cognitive defects, and the like. So these are just sort of show you very quickly, just to say these cells look like real cells, and that we can make them reproducibly in large numbers. So next slide. Oh, I can do that. Okay, here we go. So I told you we have become a therapeutics company as well, because we make a large number of cells that would have therapeutic benefit. So last year we formed a spin-out company, a wholly owned or a joint venture rather with Fuji called Optimism to treat various blinding diseases. So we make retinal pigment epithelial cells, we make photoreceptor cells that can be used for macular degeneration, retinitis pigmentosa, and other diseases of the eye. We also make heart cells. We make, so we're making some cardiac. We have both progenitor cells and mature cardiomyocytes that we're developing for heart attack or any sort of cardiac disease. And we make dopaminergic neurons. These are the cells that are lost in Parkinson's disease. And so we're producing those as well. And then for oncology, we're starting to make some of these T cells or natural killer cells. You've probably heard that Novartis and the University of Pennsylvania got approval a few weeks ago for a CAR T product. This is a patient's T cells with an artificial chimeric receptor that's been approved for leukemia. So we want to make those cells not from the patient cells, but actually make them from stem cells so that they're available off the shelf. Now you heard John talk about they do an autologous therapy. So they take the patient cells, they modify them, correct the mutation, use it to treat the patient. Linda's company started that way, but now they can use allogeneic. They can use donors, unrelated individuals. And CDI felt we were going to be a bit, I'm just going to go through here. Agnostic. We don't really know what the best starting point will be, and it will likely be different for different diseases. So for our dopaminergic neurons and for the eye products, we're making those from allogeneic cells because those are immunologically privileged areas. But most parts of the bodies are vascularized, and your immune system recognizes somebody else's cells as foreign, and the immune system is evolved to kill those cells. So what we decided to do was to generate some lines that would straddle sort of split the difference between foreign cells and self cells. And these are called, what we call these is HLA superdonors. So there are dozens of proteins on your cell that allow your immune system to recognize self from non-self. But it turns out that for solid organ transplants, there's probably 3 that are most important. They're called A, B, and DR. Doesn't really matter. But what complicates that is that we all have about 2 different versions of each of those 3 genes. So you need to find 6 matches. And that's why when you know someone who's waiting for a solid organ transplant, they can be waiting a long time. You need to find a donor that's reasonably well matched, and then you put them on immunosuppressive drugs the rest of their life. But they need to be reasonably well matched. Well, what we did was we went to the National Marrow Donor Program and looked for individuals that for those 3 genes had the same allele on both chromosomes. So we now only have to match 3 genes to a donor. So it makes it a lot easier to find matches. And this is what's shown up top here, that if you were the gray individual, you could go into those 3 individuals on the bottom, the gray blue, the gray green, the gray black, because those individuals see gray as self, so they don't reject it. But if you went into the top, you would be rejected. So what this has allowed us to do is find these, what we call haplotypes, these combinations of genes that match a good portion of the population. So we started, I told you that we announced last September we were becoming a therapeutics company, but in 2012 we started this project, and in 2013 we put out a press release that we were making these lines GMP, so under good manufacturing practices for therapeutic purposes. We saw that we were going to be there, either whether we made cells ourselves for therapeutics or we manufactured them for other companies. That's what we would do. And so we started making these cells, and the top 5 donors we chose cover about 35% of the U.S. population. So that gives us a good leg up in helping to find starting points to treat a large number of patients. And then the phase 2, which we're currently doing now, is to up those 5 donors to 200. That would cover most of the U.S. population. And then after that, we will do it for the rest of the world. This is being done in Japan by Shinya Yamanaka. They need many fewer lines there because the Japanese population is genetically much more homogeneous than the U.S. population. He needs about 100 lines to cover most of the Japanese population. We need about twice that for the U.S. So that's what we do. We are not focused primarily on rare or common diseases. What we want to do is provide the infrastructure to help those companies advance the therapy that they need, because in addition to having the smart science and knowing the medical and the biology of these diseases, you also have to have behind the scenes just the sort of prosaic work of high quality manufacturing, both in scale and in quality. And that's where we've sort of focused, sort of planted our flag and focused our company. So with that, I'm going to stop and let Maury take it from there.
So we've got, I think, about 15 minutes left, maybe a little bit more. What I'd like to do now is open this up for questions. I'm going to actually start, but I'd like to encourage the audience to ask questions as well, and we have microphones for anybody who wants it. So if you want to ask a question, raise your hand, and we'll get the microphone to you. So if I could just pick up on a theme that I think is kind of recurring through all of the presentations, which is some of the advances in cell therapy really over the last few years. And I think, one of the things we do suffer from candidly in this space is that we've been talking about cell therapy for a while, and so there's a lot of expectation that has been created. But I think we really are at a moment now, and I said this in my opening remarks, and I think you heard this today, where we really are getting close to perhaps a watershed moment where we're going to see, a number of these therapies really moving much more rapidly through the clinic, either because of the regulatory pathway, but also because of the advances in the technology. And I think that's what you're hearing from all of the 3 presenters. I mean, a lot of these folks have been at this for a long time, and Linda will tell you that they've learned all along the way. So if I could ask each of you just to comment a little bit on why perhaps this reason for optimism. What has changed over the last 5 or 10 years? And maybe John, start with you, or Linda wants to go first. That makes us really believe that we're getting closer, that we're going to see these products in phase 3 and really in the clinics for the patients.
Right. It's a good point. It's been a long time. There's been work back to the 90s at Penn that, starting a lot of the gene modified and work that, really has pioneered what's happening today. So I think you could break it down into a few categories, and I'll talk about each of them. One is, the technology is really one of the big parts. The concerns there's always been with either introducing cells, something live into the body, or a piece of gene or something genetic is, what will it do? Will it do something random? Will it park next to a gene that causes cancer. This is some of the very early thinkings that they had to work around before started creating these gene therapies. So over the last, decade or so, the technology to really be a lot more precise with this has really come around between viral vector technology, between, Tom mentioned a lot of the gene editing work that's been starting to emerge. It's really has become leaps and bounds a lot safer. They put a lot of mechanisms in these vectors to make sure that they don't replicate later. There's just a lot of technology that's been put into the safety aspect of both cell and gene therapy. So I think that's really one of the most important things that's been moving us a lot more rapidly. And then we also have the concept of, can we make these products? Our company makes them from individual patients, and there's a lot that goes into that. There's, the scale. Can we make enough? Can you dose it properly? The cells are alive. How do I even get it to the site? Are they going to be dead when they get there? These are things we actually had to think through each part of the process. And there's even technologies for that. There's manufacturing technologies that have made this a lot more efficient. Closed systems, automated. It's really just leaps and bounds than it was even 10, 15 years ago. And even when I just made talk about shipping, there's even systems now for that to make that a lot safer and a lot easier on the cells. And then in general, I think the last one is the regulatory acceptance. So back in the 90s and going in the early 2000s, when the FDA would see a cell therapy come in as an IND, which is what you do to file to go into humans, they're like, well, we have to look at this. We have to stop. We have to slow down. We have to look at this. We have to be really sure this is safe. Go run a bunch of safety analysis and make sure we're okay before we go into one patient. But you've just heard Maury mention now we're starting to see approvals. We're seeing a lot of late phase trials. So that tends to compound with the FDA, and that with new leadership now at the FDA, I really think with the programs you'll see this really accelerating quickly going forward. So.
Linda?
Thank you, John. Thank you, Maury. So I think from my perspective, the thing that is powering this forward now is a greater understanding of the mechanism of action of how cells work. So just for those of you that aren't clear, we talk a lot about cell and gene therapy, and we work here, so we say cell and gene therapy like it's one word. But really, cell therapy is when you actually take a cell that is largely unmodified. Perhaps you've put it through some type of growing system like my cells to make them different than what you actually have in your body specifically, but it's just a cell, and you use that cell to treat different types of diseases. Gene therapy means that you've actually monkeyed around with the stuff inside the cell to tell a cell to do something different. And it's a really powerful tool for treating different types of diseases because you can get new proteins made that the patient may not be able to express. So both of these fields have been clouded with their headaches. Gene therapy, many of them have been safety. We know it works, and it works fabulously, but John and his colleagues have worked for years now to try and make it safe, and they're really there. There's a lot of gene therapies out there that are safe, and it's going to change the world because it's going to actually help us make diseases better. Cell therapy is a little bit different. When we started in this field of cell therapy, and I have a slide sometimes I use, and if you go to my website, you can see it because I think it might help you. We thought that the cells that we delivered would go in and become part of whatever the damaged or sick organ was, and then it would multiply and divide and become new neighbors, and everything was going to be better. And what we learned over time is that, at least in the progenitor cells that we work in, that doesn't happen. So in embryonic stem cells, that can happen, but there's a whole lot of headaches in those because it's hard to really control what they do, right? Once they get inside the body, they might make a decision to be something that you not necessarily want them to be, like hair or teeth. And then on the flip side of that, they're strongly immune reactive, right? Our cells are immunoprivileged in the sense that they don't cause an immune response, which is really nice. And they really go in and act as a local drug delivery system, like I said, delivering those exosomes, which we now even know the RNAs and the DNAs and proteins inside that are mediating the positive effect. So that's a huge sea change, right? To go from thinking a cell's going to go in, and if we put in 10 cells, it's going to become 20, 50, 100, whatever, as opposed to we put in 10 cells, and we know that within a month, those 10 cells will be gone, but the effect of them will linger because of the exosomes and the biologic materials like growth factors and cytokines that they release. So that's why this is getting closer. We understand them better. We can deliver them in a way now that is more efficacious. We've learned a lot about dosing, and it allows us really the opportunity to provide you with a cell therapy that's going to look a lot like drugs, medicines that you've been taking or have your families take for years now.
So Linda, before we go to Tom, if you can just hold on one second, because we got a question over the live stream site, and this is directed at you. The question is, so for this particular drug, are there other muscular dystrophies that you are anticipating or thinking about for this particular therapy?
Absolutely. So one of the things that's nice about what we've learned about the cells and their biologic activity in muscular dystrophy is that it should apply to other types of muscular dystrophies and specifically other monogenic diseases of inflammation. And let me just also say that we expect to be a good partner in the sandbox with all the dystrophin-correcting therapies. One of the most difficult things, I think, in genetic diseases, and I even remember this as a graduate student, is when you see a monogenic disease, you just want to kind of reach in and fix it, because then you can make the people better, right? But there's all kinds of problems in getting that done. And one of the problems with some of the correcting therapies, like dystrophin-correcting therapies, is you probably won't get 100% penetration, which means you're still going to be left with a lot of dystrophin that breaks down and leads to inflammation. Our cells calm down inflammation. So we'll be able to partner with all kinds of dystrophies, muscular dystrophies, and dystrophin-correcting therapies as we move forward.
All right. Tom?
Thanks. Well, I think I'll just second what everyone else has said. I think one of the big advantages or one of the advances that we've seen in moving cell therapy forward is this issue of regulatory certainty. So both investors and pharmaceutical companies are largely uninterested if they don't feel that there's a well-trod pathway towards registration of a drug. And with cell therapy, the FDA didn't know what they required. They would ask you what you thought you should do. The cell therapy companies didn't know what to do. And I'm guessing Geron probably did 10 times more safety studies than were necessary, but because neither they nor the FDA knew that that was enough. One of the concerns with stem cell therapy, or starting with a stem cell like my company does, is that there's a concern that if you have undifferentiated cells after if we take stem cells, convert them to heart cells, for example, if there are any undifferentiated stem cells in there, in animal models, at least, they cause tumors. So there's this worry, how do you know that you've removed them all? You can never prove you've removed them all. You can only say you've gotten below a certain level. And it doesn't make the field look great, but what has happened with experience and time is we've wound up treating thousands of patients and haven't seen any tumors. I mean, these are just turning out to be safe. Not that we knew that was going to be the case. It was certainly, we hoped that would be the case, and it was our intent not to put patients in danger. But without that empirical data to go to investors, to go to pharmaceutical companies, to small companies like FiberCell and Capricorn don't get off the ground. Companies like CDI can't develop these therapies and take them all the way to registration. We're still going to need to partner those with pharmaceutical companies. And I think the safety issue is making both the regulators and the investors more interested in moving these therapies forward. And there are a lot of ideas out there, but it'll be slowed down if you can't fund them or you can't get approval to go into clinical trials. And we're moving beyond that roadblock now.
Great. So let me pass it over to the audience. Are there any questions here? Anybody have a question from the audience? There's one over here.
Yeah. So with the recent FDA hold on the 2 Selectus clinical trials, how would that impact the development of the therapies, or would that change how you are focusing your clinical development plans?
Yeah.
So I guess for us,
Not,
So for us, it's really a blessing, because one of the mountains that we've had to climb is to differentiate what we would sort of call, unvalidated, unproven cell therapy, which provides potentially hope to patients and families without it being validated as going through specific clinical trials with endpoints and also validation in a statistical way that this is actually working. We're treading the very traditional path, working closely with the Food and Drug Administration to give them exactly what they need to show that our cells will make a difference in the lives of patients. So we can't say enough about some of the therapies that have been used sort of in that relatively untended and unregulated fashion.
Yeah, I'll just echo that. I mean, when we develop all of our plans, and I mentioned before a lot of the regulatory statuses that the FDA has developed to give to companies developing, putting together these therapies, including regenerative medicine therapies and a lot of the rare disease. And the reason they do that is they want you talking to them and creating and designing these protocols so you have an agreement on what is going to be an effective way. We call them endpoints. What's the effective way to judge the efficacy and safety, that mostly in this case, the efficacy of these diseases. And there's just really, it all comes down to education and talking with the FDA, using your KOLs to really define what is important to the patient, what are the right endpoints, and then using their own statuses to use them to your advantage. I mean, they have something called special protocol assessment. That's something you sit down with the FDA, you actually go through your pivotal trial and design what it should look like, what should the statistics look like, what outcomes measure means it's an efficacious result. If you go in blind and do these things sort of, hoping it works and then maybe we'll just throw it over the FDA and sort of see if it sticks, that's burning, like in a public company, that's burning money of investors, that's time, that's a lot of, it affects patients obviously because it could extend approval times. That's a big problem, and that's why we try to keep conservative but working closely with the agency.
Yeah, I would just say that this is an incredibly difficult conversation to have at a rare disease meeting when you have patients who don't have any other recourse. But the importance of well-designed clinical trials can't be stressed enough. So some companies sometimes get accused of wanting to prevent therapies. Everyone believes we have cures for cancer on the back shelf, as though we don't get these diseases ourselves, or that somehow we're trying to corner the market on these things. That just isn't the case. The data that come out of well-designed clinical trials helps the next iteration, the next study, helps the next cohort of patients, and that can't be stressed enough. But I know that can be a very difficult conversation to have if there's no other options that you can offer patients.
Right. Other questions? Over here.
I just wanted to ask, for those with autoimmune conditions like myself and it targets a particular organ to destroy it to the point it can no longer function, would you then recommend both the cell therapy and gene therapy to not only repair the organ to have use again, but also prevent that autoimmune response from occurring again? And is that possible? And how do we go about getting that therapy?
Let me go first. John, you go first.
That's a
good question.
And I think it's a great question, because I think, let's just to step back to what Maury asked earlier about what do we see gene cell therapy going in the next 10 years. So both Linda, we have discussed what we call monogenic disorders, and why we bring that up is they're a single target for us to sort of just show our proof of concept, and we don't want to pick a particular disease over the others, but sometimes our technology needs to have some sort of proof of the platform before we can then go into what we call multigenic diseases, or what you just mentioned, an autoimmune disease, where you can have a very massive cascade of proteins that lead to a disorder well down the line. And do you target upstream to see if you can stop the cascade, or the sequelae of the disease? Like, that's how we pick to do with Scleroderma, is there's so much complexities and unknowns in the actual autoimmune reaction. We're going to just start by trying to help the sort of the outcome of the disease. But the ultimate goal, to your point, is, we want to do all that eventually. It would be great if you can have a construct that you could put into one of our cells that has multiple proteins, that does inflammation, that if there's a genetic component of the autoimmune disease, can addresses that. And there are technologies where that is possible, but it's in the pipeline. And I think that's something that I hope we would see as the technology increases, regulatory-wise, just proof of concept-wise, that we could start having these more cascade sort of constructs and proteins, or cells that can be pre-programmed to do that and then implanted. So I think that's an outward-looking possibility for this going forward.
Yeah, I'll just add to that only briefly that we're hoping to be able to target autoimmune disease in patients that have early stages of the autoimmune disease, so they won't reach a point where their organs are broken down, and then we'll be able to maintain what they have. And what I'll tell you is that autoimmune disease is one of those incredibly exciting opportunities in the field of cell and gene therapy, because we actually understand in many of these cases what's causing it. We just have to get in there and kind of figure out how to fix it. And so some of these is pretty close, so hold on, it's coming.
All right. We have time for one more question. Is there right there? Can you pass the microphone? Here it comes. Yeah.
My question was to Mr. Novak from CDI. There are a number of us here with inherited retinal diseases, and I know that you are partnering with Fuji and Dr. Gam for potential site replacement therapy based upon the IPS lines that you've got. When you noted all the grays and there would be less rejection for that cellular model, and you noted, I think you said that covers about 35% of the population, but that you're working to gain more IPS lines that would be able to cover a larger population. Is there a timeline for that?
Yeah, well, that's underway now, and we would expect that the majority of those lines would be available, those 200, in the next 2 to 3 years. Okay. But the good news for blinding diseases is that the eye seems to be an immunologically privileged area where we can go in with fully allogeneic cells or only partially matched cells. So we don't need to have those lines to move eye diseases forward. We just need to have GMP materials, good quality, good manufacturing practice for our starting materials. So you're in luck if you're suffering from, you're in luck, but you don't need to wait until we find your line that is a good match for you to take, to potentially benefit from these developments. And we're not the only ones doing this too. There are a number of companies working in the making these retinal pigment epithelial cells and photoreceptors. We're just working in parallel with those guys.
Just a quick follow-up. You said immunological privileged, but is not the vitreous, are there not large inflammation problems from?
It's not been shown that using allogeneic cells in the retina, either as photoreceptors or RPE, has caused any sort of immune response in the eye. So at least the cells that are being produced for those therapies seem to be generally safe, and they don't need to be autologous. In fact, there's been studies started in Japan as well using allogeneic IPS cells that have also begun.
Okay, because I know that there have been some minor infection problems with some of the AAV vectors that have been tried on an intravitreal and subretinal layer, and they end up needing to go in with steroid treatments, and then you end up having a lower transfection rate for those.
Yeah, and those should be avoidable just by proper, better technique in introducing the cell to avoid introducing infection into the eye.
Great. Okay, so we've gone a little bit over time, but I want to thank our panel for, I think, a really terrific discussion. Just a thought to leave everyone with is we've seen tremendous progress here, and I think you heard this today. I think there is a lot of reason for optimism, and especially in the areas of rare genetic disorders, and that's why we're here. All three of these people represent companies where they are working in these diseases, and I think we're going to see a lot of advances in the near future. So thanks, everyone.